So I'd like to welcome you to the first session of the National Beef Cattle Evaluation Consortium Brown Bagger webinar series. If you haven't already, I'd encourage you to visit the website, NBCEC.org. For those of you that have participated in these webinars in the past, you'll recall that we do record these and post them for later viewing on this website. And here you can also find recorded sessions from previous year's webinars as well. So I'm your host today, Matt Spangler. I'm a faculty member in the Breeding and Genetics group at the University of Nebraska-Lincoln. I'd also like to mention my colleagues, Dr. Dare Bullock at the University of Kentucky and Dr. Bob Weber at Kansas State University. As a team, we're the ones that have planned this year's Brown Bagger webinar series, and we certainly hope that you enjoy it. So we have two speakers today for this first session, both really focused on reducing the impacts of disease and parasitism. And so I'm going to just briefly introduce both of today's speakers. The first one, whose talk will focus on reducing disease susceptibility in dairy cattle via genetics, is Dr. Fernando Di Croce, who's currently Director of Global Genetics Technical Services at Zoetis. Prior to this, he earned a DVM at the University of the Center of Buenos Aires province in Argentina. And after that, he served for 6 years as head of the Department of Residency and Projects at the College of Veterinary Medicine at that university. In 2010, he completed a PhD in reproductive physiology at the University of Tennessee, where his research focused primarily on genomic technology applied to genetic improvement with emphasis on fertility and other economically important traits. During his tenure at Pfizer and then Zoetis as a senior scientist and geneticist, he served within the veterinary operations team for provision of technical support for genomic technologies through the field force and technical services team. He served in numerous other leadership capacities, and today his role really focuses on supporting the commercial adoption of genomic technologies by providing the information and tools necessary to understand the implementation and value of genetic testing services, by translating genomic research and development into solutions, and by educating both internal and external stakeholders on the features, benefits, and value associated with use of those genetics products and services. Our second speaker today is Dr. John Keel from the U.S. Meat Animal Research Center, who visit about host resistance of cattle to horn flies, focusing on genetics and phenotyping strategies. Dr. Keel is a self-described generalist. He's worked on several different things over a long, very productive career, but all those really have a focus on statistical approaches, computer simulation, and mathematical approaches. He joined USMARC in 1987, and some of his initial works were focused on computer modeling of growth and body composition in cattle as affected by different planes of nutrition and genetics background, and that really spawned the development of DESI, which some of you would be familiar with, that Dr. Keel worked on with colleagues at USMARC, Charles Williams and Tom Jenkins. In the early 1990s, he switched the focus a little bit to work on gene mapping of cattle, swine, and sheep, where his contributions were primarily database development and statistical analysis. His work, along with global colleagues, really led to the sequences of genomes for cattle, swine, and sheep, and uncovering of genetic variants within all of those species. In the last decade, he's really focused his research on genetic resistance of livestock species to infectious agents, both micro and macro, and internal and external parasites. So I'm quite pleased to have these two very talented scientists with us today to share a bit about their work. So with that, I'm going to go ahead and turn it over to our first speaker, and we can begin with the formal presentations. While we're loading up the first set of slides, I just remind the group, if you have questions, please type them into the chat box, and we'll address them at the end of the respective presentations.
Okay, can you see my slides, Matt?
Yes, I can.
Excellent. Thank you, Matt. Before you start, I would like to thank you and thank Bob Weber for this invitation. Also, I would like to thank Dr. Ken Anderson to help me to prepare and review this information for the webinar. I am really excited to have the opportunity to share some experiences with you around the genetic selections for disease traits. This is an area, particularly functional traits, that I have been working for the last 15 years, first with fertility traits, and more recently, my team and I have focused on health traits that, again, given the data economic challenges that we have today, given the animal welfare concerns that we have today, I think that working in health traits, selecting for health traits is taking a lot of momentum in the data industry today. So the contents that I am planning to cover in this brief presentation will be around, first, try to illustrate the need of healthier cows and calves that we observe in the dairy industry. Second, I would like to describe some of the procedures that we are using for creating genetic predictions for health traits. And third, I would like to discuss and introduce how we can achieve genetic progress in these traits, particularly how we are incorporating this information in multi-trait selection indexes. I will try to finalize this with some remarks and conclusions for introducing this topic. So in this slide, I am intending to illustrate the dairy cow morbidity trends for the more economic and important traits affecting dairy cattle over the years. This is a little bit dated information, but I really like the epidemiological approach that the people here in the USDA have been working on reporting this study. Particularly, I wanted to highlight that or illustrate that still our dairy farmers continue to be challenged in managing the health and the repro of their cows. If we see, in this presentation, in this trend, what we are observing is that these disorders are in the wrong direction. So we are increasing the incidence of this disease. For example, we are increasing the incidence here of the clinical mastitis, lameness, several different reproductive problems that, again, there are, for example, the last three that I mentioned are the main reasons for involuntary cooling in the contemporary and the progressive dairy operations that we work with. So again, despite of the better management and many learnings that we have over the years about this disease, our farmers continue to be challenged in managing this health and the reproductions in their operations. When we observe the morbidity and mortality in cows and heifers, we also do not observe too much improvement, particularly observing these health trends. So the risk of morbidity and mortality, I think that is higher than we would like to see. Again, this is data from the USDA, from the National Animal Health Monitoring System. Again, a little bit dated, this data. But again, as we're observing here, for example, the percent of the heifers affected with pneumonia in dark blue, the percent of the heifers affected with diarrhea in light blue, and the percent of mortality in yellow. And we are comparing here pre-weaning cows with weaned calf. Again, we show that we're trying to demonstrate that despite the fact we are trying to manage as well as possible these animals, that all the learnings that we have in this disease, dairy farms continue to face challenges in managing the health of these calves. Importantly, the economic impact of this disease is real and is really significant in this dairy operation, in these dairy cows. And again, we have some evidence here, at least several of the research that we are trying to collect in order to illustrate, again, first, the incidence rates, second, the cost per case that we have in these costly diseases, and also the increased cooling risk that we have if we have an event of this disease. I don't know, I will pick one, for example, mastitis. That is one of the more frequent diseases that we have in the dairy operations. We have an incidence rate between 12 and 40%. So as a cost of treatment, it is around $200. And again, we have a high risk if this animal experiences a mastitis event. We have a high risk that this animal is sold or die in our operation. And again, I think that improving this incidence, improving these health and wellness traits, if we include genetic selection as part of an overall management, we believe that this is a great opportunity to start with a different strategy, a novel strategy to manage better this disease incidence and definitely impact profitability in these dairy operations. So for that reason, we believe that working and developing predictions and selecting against those diseases, it is a profitable choice. It is something that it is really impactful to work in these diseases, in these dairy operations. So we know that if we can improve health, improve wellness traits by including genetic selection as part of the overall management that we need to have for this disease, we believe that, again, it is a really good opportunity, a really compelling opportunity for producers to help manage this disease and also improve profitability in the cattle that they care for. So I would like to introduce really briefly how we have developed genomic predictions for health traits, particularly in cows and in calves. So importantly, we have developed a genetic evaluation for Holstein cattle, and also we have a different genetic evaluation for Jersey cattle. I will focus most on the Holstein, but again, I wanted to notice that there are two different genetic evaluations in dairy. So this development that have the opportunity to work particularly the genetic evaluation for health traits is already published for our research and development team in the Journal of Dairy Science. I am just putting these two papers here as a reference, and I will take some moment to describe particularly some features of these papers. But here we have the description of the procedures, the methodologies, and the results of the genetic evaluations for calf and cow health events. I will highlight in these slides some of the particularly the key health traits that we are working and that are included in the genetic evaluation. So we have 7 traits that are focused on lactating cows that are on the left-hand side there. So we are predicting the risk for mastitis, lameness, metritis, retained placenta, displaced abomasum, ketosis, and male fever. These are a group of traits that we have selected in order to work with because given the frequency of these diseases, given the economic importance of one event of these diseases, so we select in order to start working with those. We also identify 3 different calf traits to start selecting for. We have calf livability or mortality. We have calf respiratory disease and calf scours. On the right-hand side, I am just illustrating a table from the paper that we just published in 2017, where you might appreciate the number of records that are feeding in the evaluation, the phenotypic means, that means particularly the incidence that we are observing in those traits, and standard deviation, again, of these lactating dairy cows wellness traits that we have in the genetic evaluation. As you can observe there, for example, mastitis. So we have, at that moment of that, when we release that evaluation, 4.1 million records feeding in that evaluation, and from these 4.1 million, 25% of those was the incidence that we have defined there. You know, for the other traits that we have, mostly we have 3 million, records, phenotypic records feeding in the evaluation, with the exception of ketosis that we have 1.8 million. At that time, the pedigree file was around 14 million animals, and we have 114,000 genotypes feeding in this genetic evaluation. So again, now this information continues feeding the evaluation. Today we have maybe 800,000 genotypes feeding in the evaluation, but at least I wanted to illustrate a little bit about the numbers about this genetic evaluation that we are running. Briefly, I would like to describe some of the phenotypic means. When I say phenotypic means, I mean the incidence of this disease, standard deviation, heritabilities for the analysis, and also, and importantly, how these traits are defined. If we continue with an example like a mastitis, so this trait was described by case per animals per lactation. So if we consider in that way, we observe a 25% of incidence of mastitis, and we have a heritability of 6.9%. So heritabilities on these health traits are relatively low, but they're ranging from 5.9 to 8.1. But again, given the experience that we have with these traits, particularly in fertility, and we know how much we can improve, how much progress we can have even with these heritabilities. We consider that they're really acceptable, and we were really happy when we have these heritabilities and for these traits. Again, importantly, each trait was defined as in this case, as a Holstein female that was recorded with the presence or the absence of the disease or disorder in a given lactation. For example, we screen a lactation of an animal. If the animal have an event, 1 event, 2 or 3 of mastitis, we consider that animal impacted with the disease. For those that they were not impacted, we consider that animal healthy. So it's a binomial kind of distribution that we have here with animals that are healthy and animals that were impacted with the disease. For example, we do not consider the severity of the disease or the mastitis. We do not consider, the pathogen. We do not consider, the quarter that was affected, just if the animal was impacted or not with that disease. This is how we define particularly the health traits. As I mentioned before, the genetic evaluation for calf wellness traits also was published early this year in the Journal of Dairy Science. I will briefly describe the number of records, incidence, and also how we have defined some of these traits in the genetic evaluation. So I just extract, a table with the same information that is in the paper, that again, in order to reiterate the definitions and how are the recording periods that we have for these specific traits. So we have defined calf livability or mortality starting with the period of the calf's life that it is not already accounted in the stillbirth. We have a stillbirth information also in the genetic evaluation for dairies. And particularly from that day to the first 365 days of the life, it is the period of that we are accounting for the cow's mortality. We believe that most of the risk of death is really early in the life of the animal for health reasons at least, and we have defined that period. Regarding to the respiratory disease, and again, we analyze multiple time periods, but based on the feedback that we have from other experts in the industry, we have defined to utilize a period from day 0 to day 365 of age. And again, I believe that we have the majority of the heifer respiratory disease challenges are accounted in that period. With the scour, basically the same. 90% of cows, heifer scour or diarrhea occur between the day 2 and day 50 of age. So we have accounted that period and defined that period as a trait. So you're observing the incidence there too. Mortality on average in almost 2 million records are around 5%, 4.7%. The incidence of respiratory disease is 21%. And again, in the scour, we observe an incidence of 26%. We are illustrating also in this table the heritabilities ranging from 4.2 to 6.0. Regarding the reporting approach that we have defined for expressing these health traits, so we have defined to report these as standardized transmitting abilities. We usually take the genomic enhanced predicting transmitting abilities that are the output of the genetic evaluation, and we transform in these STAs or standardized transmitting ability. So we define an average of 100 and a standard deviation of 5, and particularly or importantly for the interpretation, so higher values are desirable. That means that higher values represent superior resistance of the animals or the basically the disease that we are discussing, and particularly lower values represent animals that have a higher risk or they are highly susceptible to be impacted for this disease. And again, once that we generate these traits, we integrate all of these informations with already all the other traits that we have available today for selecting dairy cows, and particularly we integrate all the wellness traits with the Council of Dairy Cattle and Breeding genetic evaluation that include more than 60 different traits, including production, reproduction, longevity, milk quality, calving ability, functional type. So all the existing traits now we are complementing with the wellness traits for cows and the wellness traits for calves. And we are integrating all of this information in a multi-trait selection index that it is defined like a DWP, that means Dairy Wellness Profit Index, in order to rank the animals. But before you start describing the Dairy Wellness Profit Index, I would like to describe or illustrate that some of the studies that we have done in order to validate these wellness trait predictions in external population. As any new trait, I think in order to continue educating about this, we have developed these studies in order to demonstrate the value of this information and demonstrate also how they can utilize this information in order to progress against key diseases. Particularly the objectives of this study was to evaluate the efficacy of these wellness traits, genetic predictions in commercial herds in the U.S. hosting cows that are not contributing to evaluation, really independent and external genetic evaluation. So we enroll 3,400 cows that were genomically tested. There were, pregnant hosting females from 11 different herds in two age group, first lactation calf or heifers that were pregnant and that were experiencing the first lactations, and also some first lactations cows that were experiencing the second lactation. So again, this is information that is published from my team where we illustrate in the Journal of Dairy Science all the details of this study. But really briefly, I would like to show some of the results that we get in this study. We basically estimate the percent of differences in disease when we compare animals in the best and the worst genetic quartile according to the genomic prediction that we have for these diseases. And again, what we are observing here, it is that the best quartile groups had a 64% reduced incidence in retained placenta, a 45% reduced incidence in metritis, and a 53% reduced incidence in ketosis when we compare those with the worst quartile. That again was really exciting for us because again, when we, with the number of cows that we were evaluating here, if we see a so important disease like a metritis, for example, with just one single factor, the genomic prediction for this explaining almost half of the incidence that we are observing in the dairy, I think that was really important and demonstrating how we can improve with these particular traits. These graphs show the remaining three wellness traits with displaced abomasum, mastitis, and lameness. And again, we see important differences between the incidence of this disease. So in mastitis, we have almost half of the incidence is explained by these, predictions for mastitis resistant. So again, really important information that help us to document how important and the possibility that we have for selecting for these specific traits. So when we need to describe how to incorporate this information, this wellness traits information into multi-trait selection indexes, I think that we need to consider the ability of this index to identify profitable dairy cows that have the right balance between revenues and expenses. And particularly, we know that there are several traits that are pushing for the revenue, and again, our wellness traits, our health traits, that it will be helping us to minimize these expenses. So again, I would like to introduce at least one index that it is considering this balance between production, fertility, longevity, type, and also health traits that it will deliver, the maximum profit, maximizing the revenue and minimizing the really expensive diseases that we have experienced in the dairy industry. So, as I previously mentioned, this is the Dairy Wellness Profit Index, and all the economic assumptions that we have been using for estimate each of the underlying traits using values that using the economic values are again really consistent with what are used by the USDA. Particularly, there are a really popular index that is net merit. Basically, all of the economic assumptions of the dairy wellness profit are similar, that net merit for the core traits. And particularly for the health traits, we have been using peer-reviewed scientific literature to define what will be the economic values that will be assigned to this disease. Again, based on all of this data, and all the economic emphasis that we are included for each trait, we basically use we estimate these using the standard selection index theory. And the resulting index was, again, include 32% of emphasis in production. That include, traits like milk, fat, protein, 19% of emphasis in longevity, milk quality, calving traits, 6% of emphasis in fertility, 10% of emphasis on functional type, that include, again, other composite, body composite, the feet and leg composite. And again, importantly, we have incorporated 25% of emphasis on cow wellness traits and 8% of emphasis on calf wellness traits. We believe that this is the proper balance that we have between those traits that are generating revenue, maximizing the revenue, and those traits that are minimizing the expenses on the dairy, again, for selecting for those profitable cows that we are looking for. So some remarks and final remarks and conclusions that I would like to just share with you. First, we believe that direct selection for health and wellness traits, it is possible. For several years, we were thinking that this was difficult to do, that it is impossible to collect information, that I wanted to share with you that share experience that we have, that this direct selection, selecting for health, selecting for wellness traits is possible. Of course, we need millions of records from lactating dairy cows in order to generate and deliver genomic prediction for wellness traits. We have the possibility to develop these data pipelines that are continually flowing health events and phenotypes for the genetic evaluation. This pipeline has been established. Also, we have a single-step genetic evaluation running weekly. And again, all of this information is possible to do and possible to deliver, again, accurate genomic prediction for wellness traits. So these new wellness traits for Holstein have similar or higher average reliabilities that some of the traditional dairy genetic evaluation traits that we have, like a heifer conception rate or daughter stillbirth. So the heritability in young animals, it is really good in order to take accurate selection decisions with those. Importantly, I believe that marginal milk in dairy production is key. So mature cows give a lot of more milk than the first cow heifers. But again, in order to achieve maturity, cows need to survive and cows need to live. So productive life and health is key in order to have a good marginal milk, good extra milk, again, that will maximize our profits. I would like to share also, as I showed in the study, that wellness traits translate in a real performance differences. So these wellness traits are definitely capturing the variation that we observe in the field. So it is really important how effective these traits are in order to demonstrate the difference between the populations that we are working. And again, the overall concept that I would like to share with you is that improving this health event, improving these wellness traits through direct genetic selection present a great opportunity for our producers to help manage the disease and also improve the profitability when we couple with a really good management practice. Okay, this is all that I have for sharing with you. Thank you for your attention, and I will transfer over to John.
Yeah, that would be great, Fernando. Thank you. And while we're transitioning, there are a few questions that have come in, so we can maybe handle those now. For any of the wellness traits that you guys are considering, are any of them repeated? So do you have repeated measurements on animals?
Yeah, this is an excellent question. Yes, we have repeated measurements. So, but it is not that we repeat. For example, let's take as an example a mastitis. So, if in a given lactation we have 3 cases of mastitis, so we just account for 1. Okay, so our maybe incidence is a little bit conservative because include in a given lactation only 1 of these events that they are occurring. Now, if in the second lactation an animal gets a second case of mastitis, so that is when we are accounting as a repeated measure. So a similar animal that was impacted with the disease in the first and was impacted with the disease in the second lactation. So the answer is yes, we account for repeated measures, but again, it is across lactations that we are doing that.
Okay, another question, can maybe just in brief, visit about your phenotype analysis approach. So I think the question is particularly how have you dealt with data cleaning and modeled environmental effects, particularly as it impacts pathogen profiles and the disease traits?
Yeah, no, another excellent question. So there are a lot of editing rules that we have in our data set in order to have good quality information. All of this is produced, recorded information. And again, given several of our data editing procedures, we are, just to give some number, we are, mostly trashing 50% of all the data that we receive. So all the data that I was showing is just the data that we clean, but we receive a lot of more that we are not giving our quality checks. They are not, getting the threshold in order to be incorporated. So that again, the question regarding if there are specific pathogens that we have, yes, we have a lot of more information that we are using today. But we decide to go to the simple approach as possible. So I believe if we use pathogen-specific diseases, for example, mastitis, a Klebsiella outbreak or case of mastitis, so we will be narrowing down our data sets, and it will be really difficult to have accurate genetic predictions for that. Our strategy initially was to say, let's define just if the animal was impacted or not with the disease, regardless, if there was a severity, how many cases, what was the pathogen, and so on. So for that reason, that allow us to collect more data as clean as possible and have these predictions as accurate as possible. I hope that this helped.
Thank you. So maybe one more just very quick question. In the validation study that you presented, were differences in quartiles, were those for the individual trait or for the index? No, it's an excellent question too. They were for the individual traits. So the quartiles, the top 25 versus worst 25 are the individual traits. When we rank just for that individual traits, that was the objectives of that. But we have other data that I didn't show here where we validate the index too and see if the index, hit all the marks that the index needs to do. It select animals that produce a lot of milk, that are really fertile, and also avoid these diseases. So we are really close to publish a validation of the index in how, again, identify those profitable cows, and particularly identify those cows that are really susceptible or resistant to the diseases. Great. Thank you. Well, it looks like our second speaker, John Keel from the U.S. Meat Animal Research Center, has his slides loaded and ready to go. And so we asked John to visit about some of the work that he and colleagues there are engaged in relative to susceptibility of cattle to horn flies. So John, I'll turn it over to you.
All right. Can you hear me?
Yep. Perfectly.
Okay. So I wanted to start out just by showing you some images. I'm talking about different phenotyping strategies. And in this talk, I'm going to primarily talk about image phenotyping based on pictures. And so in this first slide, I'm just showing kind of a zoom in on a cow. I think this cow had something like 600 flies on one side, so that's quite a lot. And what we have to do with our pictures is we have to get both the ear tags so we can identify the cow and also be able to see the flies. And you can see up on the animal's top, you know, the quality of the fly picture is not the same across an image. So you can see on the top of the animal's back, there's some flies right on the top, and those are a little bit fuzzy, look like little puffs, but yet there's better detail down in the main part of the picture. And we're always kind of faced with that problem. We're trying to develop a neural network that can detect the flies and count them, but it's a little bit compromised by variation in image quality. So this is the insect that we're talking about. It's the lower left fly, and then a close up on it. A couple things I want to draw your attention to is that the mouth part is an L shape, and it's not a hypodermic needle like a thing that a mosquito has. So the fly doesn't actually poke its needle into the animal. It makes a wound with that L-shaped thing, and then it secretes proteins in its saliva that keep the blood flowing, anticoagulants. The lower picture is just showing an example, and you can see that there's both horn flies in there, and I believe those are house flies, but the horn flies are the smaller flies and the house flies are the larger ones. And the predominant flies that we'll see associated with the cows are horn flies. These animals stay with the cows during the summer, and when they get ready to lay eggs, they get up on the cow's tail and they land on the manure paths and they lay their eggs in the manure. The larvae develop after a short period of time, perhaps a few days, perhaps about a week. Then they hatch and they become adults, which the cycle continues. So I'm talking really, and I use the word resistance, which isn't maybe not appropriate. Some people, when they think of resistance, it's a all or none sort of thing. That's why I kind of changed the title to susceptibility, which could be a more continuous thing. Now, right or wrong, in terms of terminology, I use resistance as a continuous measure. So genetic differences in thrombin may contribute to it. There might be different thrombin isoforms which interact differently with hornfly salivary proteins, which are anticoagulants. There are also other proteins in the blood clotting pathway, I think a couple dozen actually, which are also potential candidates for interacting and possibly affecting susceptibility to the thrombostasin. In other words, not necessarily allowing blood flow, but clotting instead of flowing. There's the potential for immunity. There's papers that talk about both acquired and innate immunity and the role that they play and how hornflies are able to feed on cows. And then there's some nice papers on hair follicle density, which could be a function of the physical barrier of more hair or the sebaceous glands and the secretion of the sebaceous glands. There's supposed to be between 1 and 2 sebaceous glands per hair follicle. Another trait on hair that's been related to hornflies is shedding, and shedding also seems to be, so animals that shed readily seem to be less susceptible to hornflies, and there's some work that indicates that's true. So I go into more detail. I'd like to talk about this Auburn work by the Cups, and they studied salivary thrombostasin, which is in the hornfly. So the hornfly feeds by creating injury from which the cow bleeds. The anti-clotting factors allow the fly to feed on the blood. So Cup and others isolated thrombostasin from hornfly saliva. They were able to make a recombinant protein, which incidentally would not be functional in the animal because it's, it doesn't have all the carbohydrate parts that would make it functional, but it makes a very good immune target. And so they were able to make a vaccine with it. The response of the cow to the vaccine reduced the fly's ability to feed and reproduce. They actually have, flies in little cages on cows' backs, and they're able to show that the flies don't get adequate blood meals to be able to reproduce. Vaccination of cows with one particular isoform of thrombostasin had reduced impact on flies that had a different isoform. So there's some specificity to which isoform is used in the vaccine. This work has led to some attempted commercialization. I don't know exactly how far it's gone. It's called FlyVax, and they're trying to use this to market a vaccine. And then to go into more detail on the hair fly density, Stillman is a well-known, now retired entomologist, and they've found this relationship, really nice relationship, looking at Angus, Brahman, and the crosses between Angus and Brahman. So you can see that the same relationship between fly hair density and fly counts existed for the Brahman and the cross, but then the Angus cattle had the same kind of slope of the line, but then they had higher counts. And it's a really nice example of a component of hair fly susceptibility that's related to hair density. So to go into more detail on our process here, this shows a picture of a cow, an original, and we try to get the full side. The animal can either be quartered toward you or away from you as long as you can see the ear tag. And so in this case, we used annotation. We go in and we look at the picture with a computer program, and we put little dots on the flies. That's what I call annotating. And then we actually have a transparent layer electronically over the image, and it counts the pixels in those dots, and then it divides it by the number of pixels per dot. So that's how that works. This cow would have, you probably can't see them here, I guess maybe you can, but you can see that there's flies both up on the back, which is where you typically see them, but also down onto the belly, which tend to be harder to see. Then we also got the left side of the cow, and actually saw more flies on the left side than right side. We don't see a consistent pattern with that. Here shows that same cow with five pictures, and we've got both left and right side of the pictures, the amount of variation you see. Then you can see the time frame over which we took those pictures was over an 1.5 hour period in the morning. To go into a little bit more detail, I've got some zoom ins here. This is the side with the 385 horn flies, and you can see that the part on the back, the horn flies, what it looks like, and then after you annotate. And the next picture is showing down the belly, and it shows you how it's harder to see the flies. Once you get used to annotating, you can definitely pick out the flies in this picture, but it makes it hard on the software, like the neural network software we're using to try to identify the flies. You get much less contrast in your fly stringency on the belly than you do on the back, unless the lighting is just perfect. And then on really bright days, there's always a part of the cow where you can see the flies, but the cow sort of has shadows over its body, and some places it's harder to see than others. So I want to go into a little bit more on that issue of stringency of the neural network in terms of when you decide there's a fly there or not. And so there's this parameter that's related to the stringency. Basically, if the stringency is higher, you're requiring more evidence to say that there's a fly there. Lower, less evidence, so that when you have more stringency, you detect fewer flies, and less stringency, more flies. And so you'd assume that when stringency increases, you have fewer false positive flies, but also fewer flies that you miss. Yes, that's right. And then when you decrease stringency, you get more false positive flies. So I wanted to evaluate that by looking at our outcome, which would be heritability. And so that's what this next slide is. I've got those 3 different stringencies for each column of this table. And then I've got additive genetic variance, permanent environment, and temporary environment. And you can see that for the 0.7 and 0.5 stringency, which if I go back, you can see that that's detecting on average either 160 or 190 flies on average. And you can see that both of those, you get about the same heritability, the same permanent environment, the same temporary environment. But when you go to the higher one where you double this, where you reduce the stringency enough so that the average number of horned flies you detect is doubled, a lot of those flies that you're detecting are not real flies, and so that heritability drops and you get most of it in permanent environment and a lot less in genetic and in, I'm sorry, you get most of the variance ends up being temporary environment and less in permanent environment and additive genetic. So this is what I was just showing you was data from the neural network predictions, and this is data that we got from annotation. So I had a summer intern that was able to annotate 1,500 pictures on 730 cows this last summer. These are the results she got. So basically we're getting about the same heritability, actually a little lower, about the same permanent environment, the temporary environment. So what we're planning to do is like retrain the neural network with these additional 1,500 pictures and see if we improve our detection of flies with the neural network. Neural network can basically go through all of our pictures in a few hours, whereas it takes a long time to annotate. So going and reviewing some other data that's related to ours, this is actually 2 studies. The first 2 rows of this are from a producer or a seed stock producer in eastern Colorado, and the last row is data from the University of Tennessee that's using a phenotyping strategy similar to ours. They're taking pictures. That's Becky Trout, and then they're counting the flies off the pictures. And I tried to exploit the Angus pedigree and tie in the genetic connections between the 2 populations, but there wasn't tight enough relationship for that to be very productive. So the interesting thing here is for hair scores, and these are shedding scores based on 10 sons, we get about 76% heritability, and about the same amount for the fly score, which is a score between 1 and 5, with 1 being really heavy fly burden and 5 being very few flies. And then a very high correlation between them. So I'm really interested, this was based on an average of 10 sons. The people that collected this data promised to give me the individual son data, or on the sons of the bulls, and we could evaluate this further and see if this all starts to make sense. And here's some work from Central Texas. It's from ARS colleagues at Central Texas, and they're getting about 30% heritability from horned flies. This is a smaller data set, about 165 animals and about 1,500 pictures. So quite a bit of data per animal, and most of the animals have at least 8 pictures. And so still a pretty good amount of permanent environment. You can notice the standard errors are too large for the dominance and the permanent environment estimates, and so we're pretty uncertain about those actual amounts. But looks like a good bit of the variation from animal to animal is not additive genetic, maybe due to dominance, which I think is quite interesting, but it's going to take more data to be sure about that. So just to conclude, genetic difference in the cattle for susceptibility of horn flies is there is definitely there, and there's multiple possible mechanisms for genetic variation. Because there's a lot of mechanisms, I think the traits are likely fairly polygenic, which is not a surprise to most people in this group. Host pathogen interaction between horn flies, thrombostasin, and bovine blood clotting may account for some of this. Hair follicle density most likely is important. Coat color and coat pattern, there's a recent work that's being talked about a lot from Japan, where they put stripes on cows and saw a difference in horn flies on the cows just by using paint, and I think that's interesting. Hair shedding, as shown in that Eastern Colorado study, may be playing a role. In terms of future data, we collected over 10,000 pictures on over 2,000 cows, and this last summer we've got 1,500 pictures, as I mentioned before, that have been annotated. And then we have genotypes and haplotype data on most of a couple of our research populations here on the center, totaling about 4,000 animals. So I think, as Fernando actually already showed, the potential here to be able to exploit, like taking pictures to look for resistance in horn flies or susceptibility of horn flies in registered cattle is there. We can exploit the large databases of pedigree and SNP genotypes that the Angus have. The thing that's missing is we need either the breeders or other people to be taking pictures and annotating those pictures, or using neural networks to count flies. Need to estimate heritability for those and breeding values, so there's a lot that can be done with industry populations. We could identify chromosomal regions or gene associations with fly resistance. All those genotype data will help with that. Might lead to more accurate genetic evaluation. There's a system for crowd phenotyping that's or could be used as the Amazon Mechanical Turk, where you can sort of pay people on the fly for, pun intended, for annotating horn flies on images. Annotations to train a neural network to rapidly automate counting flies in images that haven't been annotated because of cost. And that's really important, because we can take a lot more pictures than we can get annotated, whether we have crowd phenotyping or not. So the benefit of a properly trained neural network would be reducing the amount of manual annotation required. It takes between 1 and 2 seconds per fly to do that. There's more skilled photographers in the world than there are skilled entomologists, and this is a way of sort of multiplying the entomology influence. Just a little bit of, I guess Fernando didn't do this, but I'd like to thank the summer interns and my collaborators. And that's all I've got.
Great, thank you, John. Just a reminder, if people have questions, please type them into the chat box. I've got a quick one here for you, John. Maybe it seems like there's two pieces to the puzzle, kind of at a high level. One is getting the fly count, so annotating that. The other, though, is being able to get the ID of the animal. And so I'm curious if you've given any thought to perhaps even using a deep neural network to do facial recognition on the cows?
I haven't thought about that, but you're saying to actually identify the cow without any kind of identification tag. And no, I hadn't thought about that. That's a good thing to consider. Actually, color pattern on cows that have variable color patterns like Hereford or Simmental, if you focus on a few points on the animal, those are fairly unique. In other words, we use that quite a bit, where we can't see the tag, but we can go back and match it with other pictures where that animal, where we have the tag and we can see all the color pattern. So the only animals that doesn't work on are solid colored animals. The other thing that works on a given day is actually the cows throw spit up on their back as part of a evasive behavior, and so that spit pattern as well as fecal patterns on the cows
Oh, right.
will actually allow you to be able to be pretty confident that you've got the right cow. Of course, that wouldn't work for pictures that are months apart or weeks apart.
Right. So Bruce Golden actually had a question asking if you could talk a little bit about the neural network topology and software, and specifically, was it a deep neural network, and if so, did it use TensorFlow?
So I have to be really shallow on this one because actually I have a collaborator that's doing that. His name is Eric Pasota. He's at University of Nebraska, and it is a deep neural network, and he uses not MathCAD, I can't remember what it is. He doesn't use MathCAD. What's the, yeah, I don't remember. Anyway, he uses statistical software that's Mathematica. Yeah, maybe that's right. So what's the one that Steve Kathman uses? Rowan Fernando, or maybe you use it too.
No, I'm drawing a blank there.
It's something math.
Okay, so a couple of other questions maybe. Is the negative effect on cow performance from horn flies, is that from the annoyance of the fly on the cow, or from the fly actually feeding the animal? And a follow-up question to that is, would thickness of the dermis affect the fly's ability to feed?
Those are good questions. I don't really know the answer. I suspect that at some level of fly burden, it's mostly an annoyance issue where the cow is just distracted from doing what she does normally. So as you probably have observed, there's a lot of different behaviors that cattle experience or exhibit with horn flies, and one of the main ones is that they group together. They sort of comfort each other, I think, by being close to each other. But I also think that just the process of rubbing against each other and sharing their tails and stuff like that, if you look at some of those groups when they're out in the pasture, you'll see that a lot of flies are in the air a good share of the time. And the other thing that I guess I'd like to say about that is I think those behaviors are effective. I think they help the cows get some relief from the flies. And in fact, one of the things that I think happens when you get a cow that's lame or that's sick in some way and she's isolated from the group is her horn fly burden will increase. And that's a good point to make, is that when you see a lame animal or a sickly animal, an emaciated animal that has a ton of horn flies on her, you don't really know whether she's got horn flies because she's sick or the horn flies made her sick, and I don't know which it is. The thickness of the skin seems like it'd play a role, but the other thing that I didn't mention in the talk is that bulls have really thick skin and they have a lot of horn flies. So I don't know, I kind of doubt that one. But
so maybe one last question, John, back to the neural network. Would you have any concern that the fly counting neural network is picking up information associated with genetics and is therefore biased?
I'm a little concerned about that, and I think there's ways of testing it. For example, color patterns are highly genetic, and so if there's certain color patterns where you cannot detect the flies, then you could get associations between the color pattern and the flies. There's also the potential that different coat patterns may be more supportive of detecting flies than other coat patterns, and those coat patterns may be highly heritable, which would then somewhat bias your fly counting. So I think those are important concerns, but I think they're also things that we can gradually tease out of the data. Yeah.
Okay, maybe one more question after that. The question is, were Bos indicus animals included in your hornfly study? And if so, are there any differences between them and Bos taurus?
Yes, big. So I don't know if you noticed on the Arkansas work, that was hair code, and Brahmin was included in that, and they saw Brahmin were considerably less susceptible to horn flies than Bos taurus. That's been documented several times. And then in our population, we have fairly small numbers of Bos indicus cross cattle, and they tend to have lower horn fly exposure or carrying capacity.
Great. Well, with that, John, I'd like to thank both you and Fernando for those two talks today. Obviously stimulated a lot of thought and a lot of questions, and that's exactly what we want. I just remind the group that our second session will be a week from today on October 16th, and there our focus will be on improving cow productivity and longevity. Again, I'll be your host, and we're going to have two speakers. Dr. Warren Snelling from the U.S. Meat Animal Research Center, which will talk about some of his more recent work related to stayability and perhaps other tools to select for efficient cows. And then Dr. Bob Weber from Kansas State is going to talk about some work that his group has been engaged in relative to the genetic evaluation of feet and legs. So with that, I'd like to thank everyone for tuning in today. As a reminder, these are recorded and will be posted at a later date to the NBCEC website. So thank you very much.